sort or another. The third category is active transport. That's where you've got something, and it's not going to be a ferry boat kind of thing. This is the bridge type structure or channel structure built into the membrane. The difference is this has a mechanism where it can use ATP energy to actually push stuff through. So it's no longer dependent on concentration differences. It will pump stuff out regardless of how concentrated it is already on one side or the other. But it does, so it's really cool, but it requires energy to do this. A couple different types. Subtype that uses ATP or light energy is called a primary active transport system. So it just drags something from one side to the other. Then there are these other systems called co-transport or secondary active transport that rely on the fact that you already may have a concentration difference with something else. Like right here, this is an example of co-transport. This is a galactoside permease. It's a channel protein that helps get lactose from one side of the membrane to the other. It's not using ATP to do it. It's tapping into a concentration gradient of protons. High protons here, high acidity there, low protons on the other side. We'll only let lactose in with the protons. They move together so that the dragging in of the lactose is piggybacking off the fact that there's already a gradient of protons. And that happens because of a proton pump, probably which depended on ATP originally. Maybe like the bacterial rhodopsin case we saw, it could be light energy. But either light or ATP energy will do uniport systems. But we have many pumps like this. Things moving like ADP moving into mitochondria or phosphate moving into mitochondria. These are this kind of system where it will co-transport. Sometimes these processes lead to charge differences. If you drag protons across the cell, you're moving positive charge outside, and you have a charge difference across the membrane. Those are called electrogenic transporters. And this is one of them. We talked about this guy before, the sodium-potassium ATPase. This is what keeps sodium high inside the cell. It's a transported, it has two protein parts, a large and a small one. Pumps out 3 sodiums, pumps in 2 potassiums at the expense of 1 ATP. So it's an electrogenic guy because it's moving unequal numbers of charges. 2 positives come in, but 3 positives go out. So you're pumping out positive charge, basically. And that leads to, since all of our cells, this is something all cells have. So our membranes tend to have a charge across them because of this. And this is the set point. It's shooting for this as a target. Inside it wants about 10 millimolar sodium and 100 millimolar potassium. 10 times more potassium than sodium. Potassium in, sodium out, ATP gets consumed in the process. This is the artist's rendition. This is a more recent X-ray structure, the real thing, with a potassium ion, the 2 potassium ions caught in transit right there. And this part down here would be involved with the energy utilization. This is inhibited, as we talked about before, by this potent inhibitor, Wabane. Just a small digression. This was used in traditional hunting in Somalia, the poison arrows. It's weird though, we have stuff like this inside our